I'm John Sterling, Editor-in-Chief of Genetic Engineering and Biotechnology News. Welcome to the special video edition of the GEN webinar series broadcast live from studios in Los Angeles. Today's webinar is entitled Biomarker Discovery, Quantitation and Analysis with Multiplex Immunoassays. Sponsored by Life Technologies, a Thermo Fisher brand, this webinar has been designed to show you how a novel multiplexed cytokine assay allows the generation of large amounts of data and therefore new analyses. The result, you will gain new insights into the roles of cytokines in health and disease. Now let's go live to the studio, and I'd like to introduce Dr. David Borden, Staff Scientist at Thermo Fisher Scientific. David, take it away.
Welcome everyone, and thank you for joining us. My name is David Borden, and I'm an R&D Staff Scientist at Thermo Fisher Scientific, specializing in immunoassay platform development, specifically for biomarker discovery and validation. Today I'm joined by my longtime colleague, Damon Cook, who is a Senior Field Application Specialist on our immunoassay team. Thanks for joining us, Damon.
Good morning, David. Good morning, everybody.
And to my left, we have one of our panelists, Jeffrey Borgia from Rush University Medical Center in Chicago. Good morning, Jeff.
Good morning, David. Thanks for having me here today.
It's great to see you. And to our right, we have G. Sharma from the University of New Mexico, Albuquerque. G, thank you very much for joining us.
Good morning, everyone. It's a pleasure to be here.
Well, it's great to have everyone here, and we thank the audience for joining today. And what we want to just remind you is that if you have questions anytime throughout this webinar, please enter them into the console on your computer screen, and we'll get to them later in the event. And so first, let's start off with the first slide. So let me start out by saying that, you know, the Human Genome Project and all of the data that's come out of that has been very exciting for those in personalized medicine, especially now in translational research. The human proteome is also very important. And now with the most recent Nature publication that came out just this past June, putting the human proteome at over 30,000 identified proteins, being able to understand how those changes in protein levels alter during different states of disease is very important. And in fact, over the last several decades, being able to better understand GPCR signaling as well as the MAP kinase cascade has been very important in understanding disease processes. A lot of new drugs target many of those molecules. So it's been very important to understand a lot of those molecules. And so what you see in this diagram on the right, I don't want to go into great detail, but the importance here, many of these early, the signal transduction pathways are kicked off many times or initiated by these extracellular responses, typically binding to an extracellular receptor, GPCR, receptor tyrosine kinase, etc. Well, being able to better measure those extracellular proteins as well as the intracellular biochemical processes is very important. And so that's the aim of a lot of these translational researchers, including those on our panel today, being able to identify specific proteins, otherwise called biomarkers. And the hope is in the research community is that these biomarkers can serve as a predictive indicator of the state of health and may actually be used to monitor or predict disease and monitor treatment efficacy. And so let's move on to our next slide. And so, you know, the aim with biomarker discovery, and let me also start by saying that one big challenge with biomarker discovery is there's heterogeneous disease populations. And by the way, I'll be referring many times throughout this talk disease. I may use the word patient or cohort. Just want to remind everyone that the tools we're talking about today are for research use only. However, our panelists today and the colleagues, many of which are watching today, you're translational researchers working with cutting-edge clinical researchers. So you're working with very disease-relevant samples. You're actually working with patients that may be battling a disease right now. So these tools are used by these translational investigators, but just to let you know that these tools that we have are designed for research use only. And so in this first slide, what you're seeing here is the classic discovery, verification, and validation model that we adhere by within Thermo Fisher. And so what you see here on the XY axis, on the Y axis is a number of antigens, and on the X axis is the number of samples. So early in the discovery process of biomarker discovery, a typical investigator is using a technology, let's say a high-density protein microarray or some other very large, high-content tool to screen a smaller number of samples, but looking at the potential of many hundreds or thousands of hits. Now, after that initial hit or the number of candidates has been identified, several or a whole team may carry out a verification process where they're looking at a subset of those markers or those protein targets that were identified, and then maybe also expanding the scope to include some of those markers that were not as statistically relevant, but just to double check. And then finally, once the verification process has been completed, an investigator will go on to the validation step, typically using an orthogonal platform where they're looking at the much smaller subset of identified markers, but using another technology that is very amenable to high throughput. So they can test thousands of samples to get very high statistical relevance, ultimately hoping to bring clarity to clinical significance. And I think, Jeffrey, that's an approach that you're taking in your work. Right?
Well, that's an important part of our strategy. In order to be able to have that sort of confidence in going forward with a biomarker candidate, you really need to change platforms. And so for our lab, we go from mass spectrometry to Luminex. And that gives us greater confidence when we're evaluating the patient specimens into will this biomarker actually work out? Because there's a lot of money involved in this sort of development. And so we need to have that sort of confidence.
Great comment, Jeff. And I think that's where we really see our expertise is in immunoassay tool builders. And let me just say right now that Damon and myself and our colleagues, and really today, let me say that I'm speaking on behalf of a lot of hard work that's been done by many of our colleagues to build these very powerful tools. But we're not oncologists or rheumatologists. We want to partner with those cutting-edge researchers that use our kits. And for example, Jeff and I are working together. We met years ago at a biomarker meeting. It's just a great example. And you'll see other examples today of how our team works very closely with the end user. And I think that's what really sets us apart, being able to collaborate. And that collaboration and working with end users really helps us build better trailblazing technologies in the area of very specific protein detection using immunoassay tools. And so let me move on to the next slide and just say that the key areas that we've focused on, and these tools, really, we do pride ourselves on building very powerful, accurate, and precise, as well as sensitive immunoassay tools. And these tools are being used in two main areas: inflammation and cancer. Now, it used to be years ago that I think a lot of clinicians and a lot of those in the field believed that there was a distinct separation between inflammatory responses and other phenomenon in the cancer realm. But really now what we're seeing is, from translational researchers, there's a lot of similarities between the two. And what we're seeing is that various forms of chronic inflammation can be a touch point or really a starting point for various forms of autoimmunity, as well as various forms of cancer like chronic lymphocytic leukemia, which you'll see an example of later in this webinar. And so it's very important to acknowledge some of these very early seminal discoveries from individuals such as Doug Hanahan and Robert Weinberg. I mean, they are leaders in the field. They're the ones, and their colleagues are the ones that really have helped identify those key relationships between the inflammatory response. And on the right-hand side, you see the classic, what some of us have called the Zodiac schematic that is really very well known by those in the cancer field. And this is the second of two review articles about a decade apart where Douglas Hanahan and Robert Weinberg outlined what's been achieved in the last decade in cancer research. And so some of the areas that I want to just point out today are, for example, tumor-promoting inflammation, metastasis, as well as programmed cell death, otherwise known as apoptosis. But the first two that I mentioned, actually, I think those are the two that we're going to hear more about from Jeffrey. Metastatic processes and metastases, as well as the inflammatory response, and how there's a relationship there, right, Jeff?
Totally. Whether I'm working with the thoracic surgeons or the medical oncologists at Rush, we evaluate each of these different hallmarks of cancer in order to try to find molecular indicators of these sort of processes going on and use those to associate with where the patient's at in their treatment. and try to make diagnostic decisions based on those, or treatment decisions, I should say. And so, yeah, this is definitely very important for us in going forward.
Beautiful. And I think that's, frankly, that's what really excites us as well, is we are building these very powerful tools. But I say this frequently when I'm on the road with you, when we talk with end users, our gratification, our satisfaction of building these tools really comes to light when these tools are being used by cutting-edge researchers to really make a difference. I mean, that's really what it comes down to, is we're really impacting human health. We're bettering human health through these tools. And so, speaking of human health, this next slide, I don't mean to, we're not going to go into details on this wire diagram. I think for anybody that has gone through, biomedical, any biology classes, I mean, it's one of these things that, especially, like in grad school, you look at these wiring diagrams and, we were responsible for memorizing a lot of this. But what we see here is the immune system is quite complex. And so, the immune system, when it works well, it's a very powerful system that really protects us from a lot of, foreign invaders. It really keeps us healthy. But when it runs awry, boy, is it a real problem. And being able to study that can be very challenging. And a lot of the inflammatory response is key. Understanding it is key to addressing a lot of diseases, which we'll hear more about today. And so being able to better measure the proteins, what you're seeing here in this classic immune profile, or this immune wire diagram, is that the various steps in the immune system and activation of certain cell types, and how extracellular mediators, growth factors, cytokines can trigger, let's say, activation of a B cell into a plasma cell, etc. Being able to measure these proteins very specifically, very accurately, and being able to measure a lot in a single sample is very critical. And so to this day, some of the most well-known affinity reagents that we use are all antibodies, pure and simple. And so whether you're using that platform, either a Western blot or a singleplex ELISA or a Luminex assay, which we'll hear a lot more about today, really depends upon your specific needs. Whether you need a certain level of sensitivity, whether you need a certain platform because of the antibody content or the specific kits that are available, whether you need high throughput. I think, Jeffrey, what you're alluding to and what you'll talk about later is once you get to a certain point where you've identified markers to get that clinical significance, you need to do thousands of samples. And then finally, in case of other needs, you might need size confirmation to confirm the size of that protein. And I think what we can all agree on is when especially you two, when you're publishing these days, reviewers of journals want multiple techniques. They want you to identify a phenomenon or a mechanism using multiple techniques that all agree with each other. And so I think that's what really excites us, is that we make tools that all help you achieve the same common goal. And so, one of those classic tools that we've all known about for over 40 years is the traditional ELISA, right? It's a very high sensitivity assay. It's a classic, the sandwich ELISA. You're capturing an antibody or capturing an antigen or an analyte that is binding to an immobilized antibody on the surface of a plate. It's a great technology. It's got great sensitivity. To this day, it's considered the gold standard. And part of my work in R&D is looking at other new technologies and seeing how we can push the envelope. But we all compare latest technologies to how well do they compare to the classic ELISA. And so the classic ELISA is great for certain things, but it does have limitations and challenges, and I think that's something that we identified early on, especially what you'll see today. If you want to measure multiple analytes, and I use today, I'm using analytes and antigens, markers, all interchangeably, but it's basically the protein that you're interested in. If you want to measure multiple in a small sample, you're very challenged because you have to use separate wells or somehow spatially separate the capture and detection events in ELISA so that you can distinguish the different signals. And what we've done at Thermo Fisher is we're utilizing a bead-based technology to really help take that forward. And many of you may have heard of this, many may not have, but Damon's going to go through it with some great detail today to really tell us how that technology can benefit us all. Damon?
Yes, good morning. Thank you, David, for that great introduction. So what I'd like to do today is to kind of highlight the technology and how it works. what we're looking here on this screen is a split screen between the singleplex ELISA and the multiplex or Luminex xMAP technology. xMAP technology was developed by Luminex, and we partner with Luminex in sharing that technology. It's a very powerful technology. So in thinking of the ELISA, as David mentioned, our goal here is to quantitate proteins, to see how much protein is in a particular sample. And to be able to look at multiple proteins simultaneously can be a huge advantage. So if you look at the screen here, you'll see this bead layout. And what this is describing to you is essentially the core of this technology, which is really taking these 5.5 to 6.5 micron beads and swelling them and then adding red and infrared dye at different ratios and allowing those beads to contract, sealing within it this particular signature, this bead dye signature. That signature now makes it a unique particular bead. It's got a specific location that we can use in it, and we'll go out in here and discuss this in a second. But what's also very unique about the bead-based system, making it flexible and flexible, is that because it's a three-dimensional sphere, it provides millions of binding sites for antibodies. Because it's a carboxylated surface, we can add those antibodies and conjugate, and each bead then has its own antibody. Well, having so many binding sites really increases the dynamic range of our assays. You know, one of the limitations with ELISA is that you have fairly truncated dynamic range. And when you go to a bead-based system, expanding the amount of binding sites gives you more power for binding and ultimately reduces or helps eliminate the need for dilution of your sample, for example. So let's move on to the next slide here, and I'll explain a little bit about how the complex works and really how you get from sample to data. So as we know from an ELISA, you have a capture antibody that's highly specific to a specific protein of measurement, right? In this case, we're referring mainly to like cytokines and chemokines and so on, but really could be any protein. Well, these capture antibodies are now immobilized to this bead as we see up here on the screen. And you can see now what we're doing is we're capturing that bead and then using the exposed epitope to bind a detection antibody. Now, what you'll have to imagine a little bit here is that there are, if we're doing, say, a 4 or 5 plex, there are these different beads, each one carrying a different bead signature from the dye I just explained. And each one of those beads has on it a different antibody. So you're capturing a specific protein, you're detecting that specific protein as well. Now, one differentiator between this bead-based system and an ELISA is really how you look at the reporter. So fluorescence is another really powerful way of reporting or determining the amount of protein over something like a colorimetric assay. Again, it helps with dynamic range, it helps with sensitivity. And so using that, what we've done is we've used something like phycoerythrin, PE as we like to call it, which is used as a reporter molecule. So if you look at the lower portion of your screen there, you'll see the complex once the assay is completed. The entire process is just like an ELISA, where it takes about 3.5 hours, so on, to run through the various steps with washes in between and so on. Well, what we do is all these reactions are happening within an ELISA well, a typical plate. Well, we use a special instrument. Luminex has 3 instruments. There's the LX200, the FlexMap 3D, which are like a flow-based type of instrument. Mm-hmm. And then they have a camera-based instrument called a MagPix. And either instrument can be used. So what we're doing is we're taking this complex and we're allowing that complex to go through the instrument. What the instrument has on it, if we're talking about a flow-based instrument, for example, is 2 lasers. There's a red laser, there's a green laser. The red laser is differentiating the different beads, and the green laser is looking at how much fluorophore is bound to that detection antibody, which is directly proportional to how much protein is bound. So you're really getting 2 measurements simultaneously. So to read a whole plate, say you're doing a 96-well plate assay, takes about 30 or 40 minutes in a typical instrument. So it really becomes powerful in that sense with multiplexing. So moving on to the next slide, here's an example, a very generic example. Say I was interested in looking at 4 different proteins, in this case IL-8, interferon gamma, TNF-alpha, and GM-CSF. Well, in a traditional ELISA, as David already alluded to, I'd have to spatially separate those or put those on different plates with different capture antibodies. Well, that leads to more time, obviously, to pipette that many samples over that number of plates. It takes more sample. If I'm using, say, a mouse sample and I only have 100 microliters, that might be challenged with trying to get that much volume to spread out over 4 ELISAs. So if you look to the right-hand side of the screen, you'll see what it looks like in a well when you're running a Luminex xMAP assay, where you're putting those beads into a well together, and all 4 of those proteins can now be measured simultaneously. So ultimately, at the end of the day, you're saving time, because you only have to pipette once. You're saving your sample 50 microliters as opposed to, say, 200 microliters. And really, one of the biggest things I like is you're minimizing variability, because you're not having to pipette 4 different times and you have variance in the pipette and all that comes with that. Everything is now captured in that one particular well, in that particular plate. So all of this is, all these proteins are bound and then quantitated against a standard curve, and ultimately you get a concentration of your proteins in samples. So I'll pass it back to David to show some practical examples of how this technology is being used today.
Great wrap up, Damon. That was a very nice introduction to the technology. And so let me just move on to a great example. What you can see here from Luminex technology is it's very powerful at looking at a number of analytes simultaneously. And so this first example was published back in about 2010 from a group over in the UK, McSherry et al. So what they did is this group used our 30-plex cytokine panel to do a retrospective study. And so this retrospective study is based on something that happened about a decade or so ago. It was a classic example in the immunotherapeutic or biological drug space. It's well known as the TGN-1412 case. And what happened was this small company decided to make a therapeutic antibody drug, and it was designed to bind to CD28. And so the aim of this therapeutic monoclonal antibody was to bind to CD28 and expand regulatory T cells, as well as initiate the release of anti-inflammatory cytokines. Well, this drug went into clinical trials, and there were adverse events, and the drug was quickly taken off the clinical trials. And there was some study of it a little bit later, but it kind of went away. And so what happened was, about a decade later, this group in the UK decided to go ahead and use our 30-plex cytokine panel to do a retrospective study to help better understand mechanism. And that's my key here, is that this is kind of an introduction to some of G's work, where she's using multiplex technology to define mechanism. And so in this specific study with the TGN-1412, the authors decided to administer the TGN-1412 drug to monocyte cultures. And then they went ahead and measured against control sets. They measured the amount of these inflammatory cytokines. And what they see in the left-hand side in that blue box, they reported about 5 or 6 pro-inflammatory cytokines, chemokines, TNF-alpha, IL-1 beta, etc. Well, then what you'll see in the right-hand side, and I'm not going to go into all the details, it's a great paper, and what they did was, ultimately they came up with this model. They hypothesized this model that they were working on. And so what you see here is that TGN-1412 actually bound CD28 of not only the cells they were hoping for, but they actually are binding to monocytes as well. That led to recruitment of those monocytes to the vascular wall, and then leading to secretion of these pro-inflammatory cytokines, but also recruitment of natural killer cells. Of course, leading to a cytokine storm, which we've heard that term before. But the point here is the authors used our kit to help define a mechanism. And this is just one great example of how our kits can help make a difference in understanding processes in human health. And I think I'll hand it over to G right now for some great examples of how her work. And just for anyone who hasn't just now joined, let me remind everyone, G. Sharma from the University of New Mexico, Albuquerque, and she's going to talk about her research program. G?
Thanks, David. Okay. So I'm here to talk about how the Luminex xMAP technology has helped me in my science. So before we go any further, I would just like to give a brief description of my research project. So I work on estrogen and its effect on metabolism. Estrogen, as we know, that it prevents weight gain and it protects against diabetes and cardiovascular disease. So estrogen, it causes these effects by acting on various tissues in the body. So it affects adipose, liver, pancreas, and skeletal muscle. So in adipose, it brings about increased fat metabolism and also it decreases adipogenesis. Whereas in case of liver, it prevents the hepatic glucose production. In pancreas, it stimulates the insulin production when challenged with glucose. And in skeletal muscle, it promotes the glucose uptake. So the net result of all these physiological effects is that it reduces adiposity, and also in combination with that, it also accelerates the glucose clearance from blood. So these effects of estrogen, as you can see in the next slide, they are brought about by 3 different kinds of receptors. So traditionally, the classical receptors which bring about the genomic responses are estrogen receptor alpha and beta. And however, recently discovered G protein coupled estrogen receptor, GPER, or previously also called as GPR30, it has been shown to execute some of the effects of estrogen in vivo. So if we give estrogen to a mouse, then you will see that we won't know whether the effect of estrogen is brought about which particular receptor. So in order to delineate these receptors, we should have proper tools how we can ascertain the function to a particular receptor. So to do so, we have some nice tools. We have the GPR specific knockout, so that when we know that only when the GPR is not present, what happens in the mice. And also we have a GPR selective agonist, which is G1, and so we know that we are specifically targeting only one particular receptor, which is GPR, and not activating the estrogen receptors alpha and beta. So as you can see in the figure, we used the plasma from the wild type and the knockout mice and the GPR knockout mice, and we saw that we ran them on a multiplex panel. So I used a 20-plex cytokine panel from Invitrogen Life Technologies, and we ran our plasma samples on that. And as you can see in the well, I have used different kinds of fonts to show what happens to different cytokines. Some of them are increased, some of them are decreased, some of them are unchanged. So if you can see the graphs, so from this particular assay, we derived our results, and we could show that the plasma from the wild type mice, they exhibited lower pro-inflammatory cytokines. However, the GPR knockout mice, they exhibited an increase in the cytokines which were pro-inflammatory, and also some other immunomodulatory cytokines were increased. So you could see that within a small well, we can measure 20 different analytes, and of the 27 had increased, 12 were unchanged or undetected, and 1 was decreased. So that's a lot of data from a 50 microliter sample volume. And since, anybody who works with my samples knows that it is difficult to generate, to get a lot of amount of sample. So it's really a powerful tool that we can actually detect so many things from such a small volume. And we published our results last year in the journal Endocrinology. And I think I would look forward to work in future with this technology to generate more data and help me understand my science better.
Great, G, thank you. So what are your next steps?
So next, since we have already established the mechanism, the phenotype and mechanism of adiposity in the knockout mice, so our next steps are to look at the source of inflammation in the mice, and also conversely, since we have seen what happens in the absence of receptor, so I would also like to stimulate the receptor with the specific and selective agonist for the receptor, which is G1, and I would like to see what happens in the obese models when we stimulate with the GPR agonist, whether it alleviates the inflammation and obesity in those mice or not.
Excellent. And what other disease areas do you think your progress will help pave the way for?
I think, as even David pointed out, and you have said, that the cytokine panel looks pretty interesting. And cytokines, they are pretty central to almost all the physiological processes. So I think if you're talking of cytokines, they can cater to a wide scientific community, be it endocrinologists, be it you can have reproductive scientists, you can have a pure immunologist there, people who are dealing with obesity, metabolism. I think anybody can help with this. Yeah.
Excellent.
It's a really powerful tool. Yes.
Great. Thank you, G.
Well, thank you very much, Gina. It's a great data wrap-up, and we look forward to seeing other publications from your work.
Thanks.
Okay, so let's move on to another real-life example of how this technology is being used, and this one is a slightly different angle. The first example that I talked about was to define mechanism. The second is to demonstrate the power of multiplexing, and so being able to do cluster analysis and really better. And I think that's something that our corporation, our organization is really well prepared for. Damon and I, we work very closely with our biostatistician team, which is amazing, and so we can really appreciate the power of this. For example, this paper here. So this interdisciplinary group from 2011 published in Blood, what they did was they used a 25-plex cytokine panel from us to help better understand the disease of chronic lymphocytic leukemia. And so what they did was they looked at about 90 or so chronic lymphocytic leukemia patients, serum from those patients, as well as about 80 age-matched controls. And what you see here is actually there are actually different groups. And so in this heat map that you see on this slide, there were a number of different people. And frankly, they distinguish them by the time of first treatment. So classically, chronic lymphocytic leukemia is considered more aggressive if it requires the first therapy in less than 5 years. And this translational research group identified those with less severe forms of the disease, of those individuals requiring treatment in greater than 5 years. Mm-hmm. And so what they did was they used our 25-plex cytokine panel to measure cytokine profiles in these patients. And what you see in this heat map, and I don't want to go into great detail, it's a very, it's actually beautiful in that how data rich. And actually, that's the power of the data richness is what we want to emphasize here. What they've identified, and you see in the blue box, or excuse me, in the black box, there's a heat map here of increased levels of certain cytokines. So let me start out by saying there's a dendrogram on the left-hand side of the heat map that is identifying the clusters of cytokines. Along the top of the heat map, you see blue, black, and yellow. And so black indicates those patients with the most severe form of chronic lymphocytic leukemia. Yellow are the healthy controls, while the blue is the group of CLL patients that had a less severe form of the disease. And what you can see here, and by the way, the coloring of the red and the blue within the heat map demonstrates the levels of cytokines either increasing, decreasing, or staying the same. And what we see here is the authors found that there is a distinct cluster of cytokines indicated by CL1. That cluster of cytokines appears to be elevated in the most severe form of the disease. Now, if you go straight below that black box, you'll see that clusters 2 and 3, CL2 and CL3 of cytokines, are not significantly changed, just very, not statistically significant, that is. However, if you look to the left and the right of that black box, you see a pink box and a green box. So these are two other clusters of those cytokines from the 25-plex that appear to be elevated in the healthier population or the less severe form of the disease. So the point is that this is a very powerful approach to help identify clusters of cytokines that may be very indicative of disease. Because, I mean, frankly, to speak frankly, we know that if being able to study human disease was just a matter of measuring one cytokine, we wouldn't be here today. And being able to look at clusters of cytokines and being able to help challenge the notion of just one marker. We'd all love it to be a single biomarker, right? But there are heterogeneous populations we all respond differently to, patient populations respond differently to medicines. We know that the biochemical, the physiology, the phenotype, the biochemical phenotype of people is different. And so biomarker discovery can be very challenging for that reason. And so using cluster analysis and the power, the very data-rich analysis here is very, very important. So looking at clusters of cytokines is one great way to identify something that is going on in a certain disease state. Another great example here is brought up by some work we did internally, and let me first say that I'm just speaking on behalf of a lot of effort from a number of people on our team. This is a really great study we did, where what we wanted to do, so let's take a step back for a second and consider lupus, an autoimmune disease that is very difficult to study, very difficult to treat. And so what we decided to do was look beyond the cytokine panel. What we wanted to be able to do was, again, try to bring clarity out of chaos, being able to look at multiple markers. So not only did we look at the 30-plex cytokine panel, but we've also utilized a new and unique 19-plex autoimmune panel that we've now launched. And so these are the two kits, the 30-plex cytokine panel and the 19-plex autoimmune panel that we offer. And we decided to use both of these kits together to help bring the clarity out of that chaos and being able to increase predictive power for a disease state. And we used lupus because it's a very challenging disease. And what you'll see in the next slide, and I'll just really keep this quick, but looking at a number of cytokines and autoantibodies, we looked at a group of cytokines, about 90 lupus samples and about 80 age-matched controls were run through our 30-plex cytokine panel. And in the upper left corner of that figure, you see we identified about 5 cytokines that are about, 98% predictive of the disease. So in other words, what that means is if you were to measure those 5 cytokines, about 98% of the time, you'd be able to predict if somebody actually had lupus. And so we were using well-curated samples for this study. So we already knew the outcome of those individuals. On the other hand, we used the 19-plex autoimmune panel, and we found out of those 19, we found about 80 markers that are, we found about 9 autoantibody markers that are about 80% predictive. However, neither one of these together, neither one of these by themselves is good enough. We all realize that a 5-plex panel or a 9-plex panel is still a lot of markers to be measuring. And what Jeff will get to in just a minute is if you want to look at increasing the statistical relevance and being able to measure in high throughput, you want to get that number of markers as low as possible. So the approach we took was to look at both cytokines and autoantibodies. And what you see here is we looked at 19 possible signatures using a unique algorithm we have. Again, great work from our biostatistics team. And what you see on the right-hand side is we identified a unique panel. I'm noting it as panel number 3. That's 1 cytokine and 1 autoantibody that's still 98% predictive of the disease. Now, the only reason I don't want to belabor the point here, but the point, what I wanted to say is that we're using the power of the multiplexing in unique ways. And so in the last example, it was cluster analysis for cytokines. Here we're looking at a panel, again, another kind of cluster analysis, but we're looking at a different kind of molecule now, not just cytokines, but autoantibodies. And this is a great segue into our next speaker, Jeff. And so, people might say, okay, well, great, you demonstrated the value of autoantibodies in lupus, but we all know that we've read in the literature, and this is a great review article from Karen Anderson and colleagues, where they outline the importance of autoantibodies in cancer work. And for any of those of you that may not have heard of autoantibodies, it's essentially, for example, a tumor protein that our immune system reacts against and launches an immune response. And so that autoantibody is a very sensitive way to measure how our immune system is responding to something that shouldn't be there. And Jeffrey's going to talk about his work, and I'm really proud to have you here, Jeff, today. We've been
thanks
working
for having
together.
me. Yes. We met at a biomarker meeting, and you've got some great, very exciting data. Jeff, take it thanks.
over.
The mission of my lab, as you know, is looking at lung cancer and trying to come up with new diagnostic tools to help the physicians when they're treating the patients. And so, as you mentioned before, these are still preclinical, but we're in a pipeline where that's the next step after we have our panels developed. And lung cancer, for those of you that may not know, is the number 1 cause of cancer-related mortality. It claims close to 1.4 million people annually. And so in the treatment of these patients, the key part here is getting accurate staging. And so lung cancer will initially be a solitary nodule, but then as it grows, it starts to become more likely to metastasize. And as it metastasizes, it spreads first usually to the local lymph nodes, and then it will disseminate systemically. And where that disease process is, it's critical for the physicians to know, and that will help not only dictate the type of intervention they're going to do, but what types potentially of agents they might want to treat the patient with. And so that's when I work with the thoracic surgeons or the pulmonologists or the oncologists, that's what I'm trying to help them gain that sort of information from the types of panels that we're developing. And the earlier, and so one key part here in the treatment of any cancer really, but particularly for lung, is the earlier we can catch the cancer, the better. And so when we can get it at an earlier stage, like stage 1 disease, the outcome for those patients is much better than if it's caught at a more advanced stage. Now, because lung cancer is virtually asymptomatic and very aggressive, it's very difficult to do that. And so there's been screening programs that have come online in the last several years using low-dose CT type scanning, but there's limitations to that, and we're trying to fill in those limitations with a blood test. And that's one of the things that I'll go on with in a second. But before I do that, I wanted to make the point that even though we're catching these tumors when it's still apparently a solitary nodule, 40% of the patients are going to recur within 4 years. Wow. Or 5 years for that matter. And that's a big portion. That's something that we need to try to improve. And so, as I said before, that we use staging to do this, and staging is done several different ways, but the most accurate way currently available is the pathologist will actually go through and look at the lymph nodes and try to determine where they're at. We're proposing a molecular-based means to do that, where as we were saying before, where we evaluate these different parts of these hallmarks of cancer to see are these different processes associated with metastasis going on, and can we find disease that can't be found otherwise. That will allow us to change the treatment for those patients and hopefully improve the outcome.
Nice.
So if we move on to the next slide, the main point that I wanted to make with this one, if you look to the left-hand panel, is when you look across for these early stage patients, the distribution of some of these biomarkers that we're looking at, we're looking for two major differences here. The first one being in terms of lung cancer screening is the difference between a benign case and a stage 1 case. And so the stage 1 cases are those two bars in the middle. And if you look at the CT image approximately in the center of your scanner here, or your slide here, that's a solitary nodule is what you see there. But what low-dose CT screening doesn't tell you is what is that? Is that a malignant nodule or is that a benign nodule? So what we're trying to do is help the pulmonologist or the surgeon make a decision on whether they should do additional diagnostics or should they do a surgery. And we do that with this type of test where we're able to go in, and if the biomarkers are changed, or if the biomarker levels are different between the cases of malignancy and with the benign cases, we're able to help inform what should happen next with this patient. And in addition to that, as we were saying before, the whole idea of is this tumor, if it is a malignancy, is it an aggressive malignancy that should be treated differently? And that's really when you get between these two bars in the middle here, with the stage 1s that don't have recurrence and those that do have recurrence. That if we can make that call too, that's added benefit for the physician as they're treating these patients. And so it's kind of a key point that we're trying to really get towards. And as we
Well, before you go on, can I just...
Sure.
So the difference of why you don't see much difference between those two groups is what you were talking about?
So probably what you're referring to is if you look at the node positives in the stage 1s with recurrence, there's virtually no difference between those bars.
Exactly.
But there is with the stage 1s without recurrence. And the key difference there is between with and without recurrence is these metastatic processes.
Right.
Now, the stage 1s with recurrence and the node positive patients, those that have the lymph nodes involved already, those are both metastatic patients. The distinction is when they presented to the physician that the stage 1s with the recurrence, ultimately, those have occult disease. They have micrometastatic disease that our current methods can't find.
Right.
here we're doing basically a molecular staging where we're finding these hallmarks and saying this patient's at high risk of developing a recurrence.
Right.
And so we should treat them differently somehow, whether it's through adjuvant chemotherapy, more aggressive surveillance, but that's the point with that.
Beautiful.
And so it's trying to add additional functionality that's, to a screening program that will help the surgeons treat these patients more effectively.
Beautiful. And that 7-plex that we've identified is very promising.
Agreed. There's, as we're talking about before, we've, have a nice panel that, we're in the validation phase now. And so it's, very promising for us as we go forward here.
Very nice.
Yes. But you brought in the topic of the autoantibodies before, and that's also been an area that has been traditionally an area of great interest in the field and in our lab as well, and we have interactions on this area as well. And the idea here is, as you were talking about before, with the lupus patients, being able to have a second type of biomarker, in this case an autoantibody, where we're measuring, an immune response to the tumor. Or on the opposite side is if there's immunosuppression going on. Can we see that? Because we all carry a certain amount of autoantibodies, and if all of a sudden those are going down, that's also indicative of a biological change. And so by associating those with a disease state, as we're showing in this heat map here, that with these different groups that we examined, we can actually see clusters of changes going on associated with different histologies even. We're seeing differences between adenocarcinoma and with the different types of non-small cell lung cancer. And so this is a very promising field that as we integrate those in, I'm thinking that's going to get us closer and closer to where we want to be in a diagnostic platform for these patients.
Very nice, Jeff. And I think this is a great example of how we're using our technologies in established ways and very unique ways, and being able to really push the limits of how these technologies can make a difference. That's wonderful, Jeff.
Thanks.
And so I guess to move forward just for a minute, what we'd like to do is have Damon talk a little bit more about, now we've seen some great examples of how these technologies are used and out in the field, but these kits, it's not trivial to put them together, right? I mean,
right.
It works beautifully when it's received. But building that assay, and I can appreciate that as a tool builder, an assay tool builder, it doesn't come easy. And there's a lot of work to ensure that quality and making sure that things don't cross-react, all that. And Damon, you'll go into that detail.
Right.
It's very important.
Yes, absolutely. First off, thank you to both G and Jeff for sharing excellent research work you're doing. And what I thought I would do is just spend a few minutes explaining to our audience really how these kits are put together. I mean, we talked a little bit before about the antibodies and the beads, and we've talked about the instrument alone, but now we're going to just take the assay kit portion of it, and we're going to dive a little bit so you can understand the process. If you're in the audience and maybe you're developing assays for different technologies or whatever, and these are some of the steps we use, and maybe there's some similarities there, but we've honed these skills over the past many years, and we've really come up with a process that works very well, produces assay kits that are consistent and provide great data. So in looking at your screen, you'll see they're basically a flow. Just going to talk through a few of these points. The first is really coming up with a plex selection that makes sense. A lot of our panels that we offer as a company really are designed by our customers, designed by the end users. With Jeff and G explaining kind of what they're doing and their input, we use that a lot of times to build our assay kit. So really, it's a reflection of what the research community is doing. And a very, very big point right up front when you start making an assay kit is you have to establish good, realistic specifications, and specifications that really are going to translate from an assay design all the way through to a finished product that's going to work the way you intended it to. And we've talked a little bit about antibodies, but one of the things that really the building blocks for a good multiplex tool is to have great antibodies, especially with capture and detection, and making sure that they're accurate, precise, and really giving you specific data. That you're not getting, we'll talk a minute about crosstalk, you're not getting the wrong data, essentially. So we have you know, Thermo Fisher has grown, we've got a wealth of resource. One of which is we have a humongous amount of antibodies. We have Abfinity, which is a great type of antibody that we can use, recombinant antibody. We also have excellent resources for monoclonal, and we prefer those types of antibodies over polyclonal. We find that we just get better performance, better consistency between lots. A good buffer system, great standards that have been properly calibrated, and you know, conjugating the beads with those proper antibodies, all a big part of this. We also spend time once we've identified those antibody pairs to really try to titer out the detector antibodies so that we get good measurable amount of protein in that dynamic range I referred to earlier, but also that we're maximizing sensitivity. We want to fill that dynamic range up as much as possible and get the lowest possible detection we can on the low end. So one of the biggest things right there in the middle of your screen, you'll see crosstalk. This is not a study that you would typically do with a singleplex ELISA, but with multiplexing, it can be a challenge. You can start out well, you've got your 10 targets you're looking for, and then you want to make sure that the antibodies aren't seeing the wrong protein. And you may get into this process and realize that an antibody pair is cross-talking, which just means that antibody A is seeing protein B. We have a variety of ways of testing our detection and our capture antibodies to those proteins, and then that'll be a feedback loop back up to maybe the second box on the screen there, to maybe looking at a different antibody pair. So we're constantly massaging that to make sure that we get the right antibody pairs from the beginning and that they're validated through the process. We also test natural sample. We want to look at plasma and serum and supernatants, and we oftentimes will do application notes that will look at various things like lysates, homogenates, and different samples that our customers might be using. We also look at spike and recovery. We want to make sure that when we add a certain amount of protein to our sample that we're getting that recovery back out, that we're not getting a false elevated value or something that's somehow suppressed. And then the big question everybody asks is about sensitivity. You know, how sensitive are these kits? And you know, I would say that on average these kits can be a similar performance to that which you'd expect in an ELISA. Sensitivity ranges a bit on the low end, but depending on the assay, we see sensitivities less than 1 picogram per mil, up to 10, 20 picogram per mil, depending on the target. One key point, though, is back to precision. That as we build these assays, that we make sure that our CVs, the variance between samples, between pipetted samples, even between plates, is good, less than 10%. So, as a user of the 20-plex, you've seen some of these, quality control points that we build in. How does that translate to your needs at the bench?
I think it's very important for any researcher to have a thing that works, that is reproducible. And since I work with mice, I work with in vivo data, and I think as Jeff would also agree, because he also has a lot of variability within the samples. So I would say that we know that already the variability comes from the sample, so it's important to ensure that the kit does not introduce additional variability and we don't include that factor.
Excellent.
So yeah, I think it's very important for this, what you're doing. It's great, I think.
Yeah. And as part of what I do and what David does is we're constantly talking with our customers. We want to hear how these kits are performing in the field, and we have a direct connection to our business teams, operations, R&D. We have that open line of communication, builds a strong team. Once the kit is designed and we've validated all these steps, we will then go ahead and do the bottling step, package the kit up, and provide these kits. And lot sizes can vary depending on demand. But we have several panels in our inventory. We also build custom panels too. So if there are different targets that people want that aren't necessarily maybe of one panel, but they want to mix and match a bit, we have a tool for that as well. So a little bit more into how we want to support this in the field. We, as a field application specialist, one of my biggest joys of satisfaction in this role is to really be able to see this in action. And we've seen that today. We've discussed that. But I'm part of a team of FASs that cover the globe, that really want to make sure that these assays are run correctly. If there's any issues with training, I'm sure there may be questions out there already about software, about instruments, and where can I run this assay. We can help with that. David and I work together in a scientist-to-scientist program where we do seminars similar to this webinar today. We'll go to your institution, we'll share our experience, and we want to hear what you have to say. We love questions. We want as many questions as we possibly can.
Absolutely.
There's an email there. You can send us an email to the Luminex FAS at lifetech.com and ask us your questions. Once users get into these assays, they might find that there's some troubleshooting needs. Maybe the instrument's not performing well, or maybe there's a specific sample type that needs a bit of work. We'll help troubleshoot, go through data sets, and really help you analyze your data. It's not just following a procedure in the kit, but it's actually looking at the data and trying to make sense out of it. And we really want to do that for you. So we're really here for you. We want to provide the best possible support as really a whole team. So I'll turn that back over to you, David.
I think you summarized it beautifully, Damon, and I think that's the key. Is we just want to also demonstrate that how close we are connected to our end users. And I think we've worked together for a long time, and it's one of those things that we really do pride ourselves in having that connection with the end user. And so, before we take our questions, and by the way, let me remind everyone again that if you do have questions, please enter them into the console on your screen. But before we take those questions, let me first of all say that this webinar will be archived, and you'll be able to view it later. But first of all, Damon and I would love to thank our guests, Jeff and G. We really appreciate you coming out today. It's fun speaking to you about your work. It's going to be great to see advancements and publications. So thank you very much.
Thanks for having me.
Yes, it is completely our pleasure.
Wonderful. And next, we'd like to thank Genetic Engineering News, Genetic Engineering and Biotechnology News for their support of this. We would like to thank everyone that's been joining us, all the viewers. We really do appreciate the time, and we hope that actually this is just the first of many conversations, like Damon was saying, the first of many conversations we'll be having with you. So now we have a couple minutes. Let's go ahead and take some questions from the field. I think we have some of the questions that people have been asking about. Looks like is the assay quantitative? I think we've talked a little bit about that. You said that the standard curves are very large dynamic ranges, very sensitive. It's very important to note that these are highly quantitative kits.
That's right. And let me just add to that question. Quantitative is important. I've seen some questions come in regarding sensitivity, lower limit of quantitation, a little bit about software. Obviously, we've mentioned that the instrument is a key piece to this technology, and on that instrument there are software systems that's really able to take the data from that plate and go through and classify the bead, and as we talked about earlier, but then also run those into standard curve. So, for example, if I'm running a 10-plex and I'm using it in a 96-well plate format, what I'm going to end up is getting 10 separate standard curves. I'm not running 10 different lanes. All of those standard curves are coming out of that one lane because, remember, we're multiplexing. So each of those beads are represented there. So ultimately, 10 standard curves gives me 10 sets of quantitative data. The way you can think of it is if you're running an ELISA and you get 96 data points, well, if you're doing a 10-plex, you're getting 960 data points off that same plate, all quantitative, typically in the picogram per mil range.
That's great. And you know what? Another question somebody was asking was, how does this compare with ELISA? And I think you addressed that it's quite comparable, right?
That's right. I mean, we'd expect to see data, again, in the picogram per mil range. The one big advantage, like I mentioned earlier, over ELISA is that the extended dynamic range gives you an additional log, usually. Typically, ELISA is 0 to 10,000 picogram per mil. We'll see Luminex hit 10,000 20,000 plus on the top end of the standard curve for concentration. So it can really allow seeing more data and less dilution, which ultimately less real estate's needed on a plate. Again, another cost-saving measure.
This is another great question. So somebody was asking, do you validate the entire multiplex assay kit, or do you validate each antibody before you put them into the kits?
Yeah, that's a good question. So, I mean, it would be challenging to just validate the endpoint. I think it's very important to qualify each reagent as you go through the process. And so each of these antibodies that we use are qualified, are checked and validated, and the steps that we lined out earlier really are there as checks and balances. We'll get to a step, and again, it's designed as a feedback. So if we get to a step where we realize that maybe that's not the ideal antibody pair, we have something in line ready for it. So we have a depth chart, you may say, to feed those reagents in there if needed to make the best assay. But yes, and I would just say this about the validation: it's not just a one-time process. I mean, we're constantly retesting these kits, each lot, and each lot has its own data sheet that has specific data points to it, like recovery and sensitivity, precision, and so on.
Well, that's a great question, a great answer. Thank you very much. Another question, actually, for the panelists themselves, for Jeff and G. Compared to other technologies, other than the multiplex, what made you decide to choose these assays versus other multiplex tools?
I think for me, the thing was that I had a very limited sample volume. That was the one thing. And the other thing was because I had to rule out ELISA, as Damon pointed out earlier, that you cannot use 200 microliters. I would just get like very few, like just 300 or 400. If I use it for this, I cannot measure other things which I need to measure. So I think that was the limited sample volume was one. And then the other point was that I wanted to run this screen and get the cytokines that interested me. And then now I would validate them using other technologies as well. So I think most important was that I can get a lot of data from a very less sample volume, and then I can validate those later on with other technologies. So I think that prompted me to pick up this particular technology.
And that reminds me of another point. I would just add that we also want to correlate these assays to ELISAs. I mean, this is certainly not an anti-ELISA webinar by any means. ELISAs are certainly a valid piece.
Oh,
and
yes.
A lot of the times, Luminex can be used to screen, and you whittle that down to one or two targets, and ELISA is a choice, a very good choice.
And I think ultimately when you want to confirm, so say for example, because in a Western blot, you know for sure because you're also looking at the molecular weight of the protein as opposed, and also the levels. So you can screen with this, and then you can confirm with other ways, say for example, an ELISA or a Western blot. So I think they will all go hand in hand very well together.
Yeah, and that occurs at the development stage too. I mean, we're using ELISAs and Westerns to develop and to qualify antibodies and look at these various interactions of these reagents.
And yes, you can be more confident about your data when you are doing that because you get the same result from 3 different platforms. I mean, and then you put it in the paper, it goes to a better journal, referees are happy, everybody's happy in the end.
Yeah, and that correlation data to ELISA is provided in a lot-specific data sheet. So when you're looking at that information, you'll see the correlation information and see how well the two assays work together. So you can transition between singleplex and multiplex with ease and with confidence.
That's great, and actually that. Oh, I'm sorry.
Yeah, no, actually, I wanted to add in that one of the other reasons that we kind of selected Luminex is it's already in the clinics. We're trying to select, develop these, new panels ultimately for clinical applications, and we don't want to select a platform that requires all the hospitals across the country to all of a sudden buy a new instrument. We want to use something that's already there, and a lot of institutions already have Luminex. And so many HLA labs already use it, micro labs, they use it. And so it makes sense to stay in a platform that's already used. So
that's a great addition, a great comment. I mean, specifically, as we're looking at new technologies, because we're constantly sensing the across the spectrum to see what new technologies are out there, what new technologies we can develop. And I think you've hit a very good point that having instrumentation that's already well established is critically important, right? It's been out, it's trusted, it's respected, and so very good.
FDA approved.
Absolutely. Very good comment. We have a little more time, so let's see. There are a couple other questions here. Somebody asked what kind of software is being used to analyze these data?
Right. So there's really 2 stages of the way I look at it, the analysis. Obviously, the data acquisition on the instrument and the analysis of the standard curves. The software that's out there now is very robust. Exponent's a great program. Allows you to go through and look at each curve, validate the data points on that standard curve, identify your LLO cues, and so on, and then be able to generate a really nice spreadsheet of data. The second stage of and this is more maybe for you guys, when it comes to looking at like that cluster data, there's got to be, statistical programs, analysis software out there that's beyond just the Luminex platform that allows you to take data not just from Luminex but from other areas and collate that and put that all into, like you showed with the autoantibody data.
Absolutely, and we work very closely with our biostatistics team. You guys out there, you know who I'm talking about. You guys are great, and so we really do appreciate the knowledge and the power that they bring to our team as well. Yeah. Great comments.
I just noticed a question about intracellular markers and about what kits we offer. So I'll just take this moment to just kind of identify kind of what we cover. Maybe it hasn't been clear. I mean, our area of focus is really around extracellular markers, proteins, things like cytokines, chemokines, growth factors, and so on. We have kits for human, monkey, mouse, rat, swine. We also have intracellular markers too. We have an AKT panel that's very popular, looking at phosphorylated proteins in the AKT pathway. We also have ELISA kits too that cover many of those intracellular markers. So we understand that it's important not only to look at the outside of what the cell is doing, communicating with other cells, but also looking at pathways and cascades to see how that nucleus is getting that message from the receptor.
Great comment. And we just got another question. Somebody was asking, can I choose which proteins to measure per well, or are the panels preset? I think that's a topic of our custom panels.
Yeah. So maybe I can help. Maybe direct this at you, G. You've run the mouse 20-plex, and you've identified. I mean, it's kind of the key scenario. You've identified these key targets that you like. So one of the things we do is obviously provide off-the-shelf kits like the mouse 20-plex. But we also have a custom channel too. So you find it advantageous to have options. So if you run the mouse 20-plex and you say you identified 3 or 4 targets, like you did, being able to go to a custom blend of those targets is an advantage, true?
Definitely, because especially now, because I have measured those cytokines in the blood, but I don't know what is the source of those cytokines. So when I start looking at individual tissues, then it would be better. Then I don't have to run all the 20 of them, so I know that these 7 change. So I would just go and order a custom panel and then get the extracts from the tissues and then maybe look at the cytokines that, okay, this is increased or this is decreased over there.
Right,
so I guess I can try to do that. And I think it will be really helpful that we also have a custom panel.
Yeah. And again, to clarify for the audience, when we refer to customs, we're referring to a custom blend of targets that we already have. We're not necessarily referring to custom development, although we will collaborate on occasion with folks to develop new panels. Like I mentioned, with that a lot of our panels really are working with customers to identify key markers.
So great comment. We had somebody. This is a great discussion point. Somebody asked, what is the value, and I think we've already covered this, but what's the value of looking at a different technology between discovery and validation? And I think, Jeffrey, you hit the nail on the head with talking about your mass spectrometry work, as well as using another platform. And I think this question was kind of geared around, is that truly of value? And I think the work that you've shown before and now really does bring that to light.
Agreed, and I think that unless you have unlimited dollars, which these days nobody does, the whole idea is you're trying to keep from following a lead that doesn't work out for you.
Right.
And so by changing platforms, that gives you a confidence that molecule is actually changing in concentration associated with some biological phenomena or however you're looking at your system. And if you don't do that, you run the risk of following these biomarkers potentially through several rounds of validation before you get to the point when, with larger populations, potentially multi-institutional, that, hey, it doesn't work all of a sudden. Well, why is that? Well, if you change platforms, you gain a confidence that this is, in fact, actually changing in our sample. And, that gives you the ability to go forward with more confidence and really economize your studies, too, as opposed to, chasing down, 100 leads and only 2 of those work out. You can narrow that down to say 10 leads and follow those, and it's a much better cost-benefit ratio. So
absolutely, I think I couldn't have said it better. I believe the same thing, that really, besides the economy state of it, it's really all about being able to identify those markers that will be identifiable by many people, right?
Mm-hmm.
Because early on, people worry, in a specific study, you worry about artifacts, data artifacts that are maybe representative of a single technology. Whereas if something is truly going to go into the clinic, it's something that we really want to be able to identify no matter who's doing the experiment, how it's being run, the precision. I mean, of course, we're doing everything we can as accurately as possible. When it comes down to a clinic setting or something else where thousands of people are doing this, we want those markers to be robust, right?
Agreed.
Yeah, that's great. Okay, well, I guess we have only a couple minutes left, and so we want to, again, thank you, everybody, for their participation today. Again, this webinar will be available for streaming download later for some time, and we really do appreciate your participation today. And again, if you have any questions, you see on that last slide the Luminex FAS email address, as well as our contact information will be available. So again, we hope this was the first of many great conversations we'll be having with various scientists in the U.S. and around the world. So we look forward to carrying on these conversations at the bench. So we'll see you at the bench very soon. Thank you very much, and have a great day.
Thank you.
Thank you.